talk more about other mechanisms underlying cancer. So I told you about oncogenes. Those are genes that play very fundamental roles in cellular division. They are like gatekeepers responsible for timing behind cellular division. So when their function is increased, when their activity is increased, then the cellular division also increases, and that leads to uncontrolled cellular proliferation, tumor in cancer. So when this proto-oncogene mutates, this is typically, and when it's involved in cancer, typically this is a gain of function effect. And we talked about these major functions of proto-oncogenes. So they encode growth factors or factor receptors, growth factor receptors, or they encode transcription factors, or they are encoding proteins that transmit some stimuli for further cell division. So any of these major properties, functions, can be magnified through mutations, but these mutations happen at very specific sites. So that's why it's relatively easy to distinguish TSGs or tumor suppressor genes from oncogenes, because tumor suppressor genes accumulate when they mutate. The mechanism is comparable with the analogy to brakes and the cars, that there is no brakes on genes that they control. So these mutations can be taking place in any sites of TSGs. So those are typically inactivating, truncating mutations that inactivate tumor suppressor genes, which is again different in comparison with oncogenes. Proto-oncogene activation may occur through a number of mechanisms, and again, that leads to some superactivation of the gene. So we may have an abnormal protein. So those originate through very specific mutations that increase their function. It could be also through regulatory mutation, which leads to some excessive amount of protein. So this effect is similar. So this abnormal protein is super active, but there might be the structure of the protein itself may not be modified, but there is more of the product through regulatory changes. Then there might be translocation that leads to some novel chimeric protein that is more active compared with the original normal protein, also leading to oncogenic activity and properties. And finally, gene amplification, which is like gene duplication, that this particular proto-oncogene is duplicated. So now, instead of one copy, it may occur in 2 or 3 copies. So there's an excessive amount of the protein, again leading to this abnormal activity and gain of function result. Now I'll show you tables with lists of oncogenes and TSGs. I will emphasize those that are important. I mean, all of them are important, and it's by no means a comprehensive list of oncogenes. So there are more than like 80 of them, but I just listed a dozen or so. The question, I guess your question is, which parts of the table you need to remember? So definitely you don't need to memorize the chromosome location of these genes because I don't remember it either. Although in the medical program, it's not required either, but I saw some board exam questions that actually use this information about the location. So if you remember the location, then it would be easier to answer the question. But it just, I'm including it for completeness. So just to give you a sense where these genes are across the genome. Okay, so ABL is a gene underlying chronic myelogenous leukemia, CML, and I'll tell you more about CML, which means that it is an important oncogene that you want to remember. I'm sorry. So in other words, if one of these genes pops up again during my lectures today, it means that we pay more attention to it than others. Also, another thing is that you can see when you look at cancer that these genes are involved in, you will notice that some of them are more specific, some of them are less specific. So those less specific underlie many types of cancer. So of course, it's not easy to, or I won't really require you to know like what all types of cancer that they cause. But there are some like this, for example, EGFR or ALK and ABL. Those are more specific. They cause just a certain type of cancer. So they're not very promiscuous in a sense of like underlying many types of cancer. Does that make sense? So it's easier. So and this is also what I've noticed with reference to board exams, that there are many questions, like for USMLE in particular, but also for COMLEX, there are questions about genes, but they tend to concentrate on those that are very specific in terms of the cancer that they cause. Like I said, you want to remember ABL from this table, probably ALK. Please remember, try to memorize as much as possible from these tables. That's bottom line. But like I said, it's easier to remember those that are more specific. You want to definitely know this MIC or CMIC, the different conventions of naming the genes. So that's another thing that you want to remember. Old conventions and new conventions, because you never know who is writing the questions for exams for you. So this MIC is a transcription factor that promotes cell proliferation and DNA replication, and it's involved in leukemias and lymphomas, including Burkitt lymphoma. So the Burkitt lymphoma is something that shows up in board exams a lot. So I'd like to bring your attention to it. And then NMIC is also very characteristic. The N in front of it stands for neuroblastoma. So it's a similar gene to the previous one, involved in cell proliferation, but it underlies neuroblastomas, retinoblastomas, and lung carcinomas. Then this RAS-H or HRAS is also quite unique because it's involved in bladder carcinoma. And then CRAS, we talked about CRAS, and we're going to talk about CRAS more today. So this is a super important gene, even though it's not very specific to types of cancer. It's quite promiscuous in this sense. We mentioned this PIK3 K kinase. This is a kinase with phosphorylation activity, which is involved in breast and cervical cancer. So it's another one that you want to remember. And then receptor tyrosine activation gene, or RET, is involved in medullary thyroid cancer. It's another gene that has some very interesting function as an oncogene, so it's worth remembering. And finally, TERT, or H-TERT, which is a telomerase that maintains chromosome ends. It's the enzyme that extends telomeres at the end of chromosomes. And it turns out that it's involved in like in brain cancer and other central nervous system cancers, but also bladder, thyroid, and skin cancers. So I mentioned KRAS, and I showed you its role in like colonic cancer before. So this is one of the first gatekeeper genes that mutate in cancer. Turns out that one type KRAS, or non-mutated KRAS, as a proto-oncogene, cycles between an active GTP band and an inactive GDP band state. So GTP is guanosine triphosphate. This is the nucleotide which is incorporated into RNA, but also functions as a storage of energy used for translation. So this GDP band state predominates, and it occurs mostly in inactive state in non-dividing cells. But when it mutates, when it KRAS mutates and underlies oncogenesis, then it's continuously in this active GTP band form. So it's shown here with this arrow. So we see that in the normal cells, the arrow is thick, leading to GDP, which is the inactive form, versus cancer, when this KRAS is this GTP, the active band form for the most part. And there is a related cascade of responses, biomolecular, biochemical responses to this activation. So there are some markers that can be used as cancer markers. So for example, there's some cell signaling receptors that are activated, like CRAF and this PI3 kinase, and they upregulate GLUT1 glucose transporter, which is also used that leads to increased amount of glucose, which is important for the cancer, and it can be used as a marker for some types of cancer. Does that make sense? So this, I would like you to remember these two states of KRAS. So here it's RAS, but again, this is the old convention, how we write it, so you need to be aware of both. There are more related genes like that. So it's like a RAS family, but in this case, we're talking about KRAS of KRAS. And I want you to be aware of these markers, like CRAF and the kinase and GLUT1. This is RET, which is also an oncogene from the table. It encodes a cell surface protein, which is the receptor that contains a domain that binds signaling a molecule. So this is the external part. So this is the receptor that binds some ligands. But it has also intracellular domain, which is this tyrosine kinase domain. And you need to know that tyrosine kinases are a class of enzymes that phosphorylate tyrosines in proteins. And this tyrosine phosphorylation initiates a signaling cascade for many biochemical reactions involved in signaling between cells, but also within cells. And so in other words, this is like activation of proteins. This phosphorylation is required for many proteins to be functional. So these mutations in this red gene cause increase of this kinase activity, even in the absence of the ligand. So the ligand is no longer required for this receptor to be active. And then once this receptor is active, also this kinase, tyrosine kinase is also active, and that recruits multiple proteins, activates other kinases, leading to super active cascades and biochemical pathways. One of the mechanisms that I emphasized leading to transforming a proton oncogene into a oncogene is translocation. And here is an example. This is chronic myeloid leukemia, or CML. And the gene underlying it is ABL1. It was one of the first listed in the table. And I told you that it's important because the mechanism is very interesting of activation. So it turns out that this ABL1 gene, normally located on chromosome 9, can be translocated into chromosome 22 to a specific site corresponding to another gene, BCR. So we don't exactly know what BCR does, but we know that when ABL1 translocates, then it lands in the neighborhood of the gene, and they are both translated as a chimeric protein, as a novel protein that is more active. This fusion of BCR and ABL1 still contains the active ABL1 domain, and turns out that in this chimeric structure, activity of the gene is higher compared with the regular gene, which gives the construct, the transcript, oncogenic properties underlying CML. This fusion, this chromosome fusion is known as Philadelphia chromosome. And so it was relatively easy to notice because of the chromosomal translocation as an underlying mechanism of CML. So that led to some interesting developments in pharmacology and cancer treatment, because based on the mechanism of kinases, tyrosine kinases, we could design drugs that work as inhibitors of these tyrosine kinases. In this case, as a inhibitor of this BCR-ABL kinase. This drug, this compound, replaces the ligand and is more easy to bind with the kinase than the substrate itself. So that leads to a quite effective inhibition of this ABL1 kinase activity. And there are more drugs like that. So that was imatinib, but there are more drugs like that. You see that most of them actually target tyrosine kinases. There are some other kinases that also like serine, threonine kinase inhibitor, but the mechanism is similar, that they replace the original ligands. Some of these drugs have higher affinity to these kinases than the original ligands, and that's the inhibition result or effect. With except for breast cancer and this drug, the famous one, trastuzumab, which is based on monoclonal antibodies that actually bind to the receptors themselves in the cancer. So that was about oncogenes. Now this table shows you TSGs. The table is shorter, so I would encourage you to memorize them all. Again, with the exception for chromosomal location. But here's an interesting feature of these genes. Namely, they occur in either familial or sporadic form of cancer. And it will be clear in a moment why that happens. But it's something to remember that first, these genes were inferred in some familial cases that in many instances actually had their own names as syndromes. So for example, a p53 gene was implicated in Li-Fraumeni syndrome, which is heritable within families, quite dramatic symptoms. But in sporadic cases, we know that p53 is involved in lung cancer, breast cancer, and many others. APC, I showed you APC was one of the first gatekeepers that mutates in chronic colorectal cancer, so it's also important. It also underlies some familial, sometimes we call them Mendelian or hereditary types of the syndromes. And then VHL stands for von Hippel-Lindau syndrome, because this is how it was identified first, but turns out that it's involved in clear cell renal carcinoma. And famous BRCA1 and BRCA2 that I'm going to talk more about in a moment, related to breast cancer and ovarian cancer. And finally, MLH1 and MSH2, those are DNA repair enzymes underlying Lynch syndrome in the familial case and colorectal cancer in the sporadic cancer type. So let's talk about p53, which is very important gene because it functions as the guardian of the genome. p53 stops replication in cells that have some DNA damage, and it targets the unrepaired cells for apoptosis. In other words, DNA damage in cells stimulates production of p53. So this is the normal function of p53, like all those listed. It also stimulates production of p21, which inhibits cyclin CDK complexes and stops the cell progression and cell proliferation. It stimulates DNA repair enzymes like GAD to fix the DNA problems. And if DNA is not repaired after all this activation, then p53 activates two genes for apoptosis, BAX and IGFBP3. You have all these major functions here. So that's that illustrates how important this gene is, and when it mutates, why these functions lead to cancer. So this is p53 overexpression in tumor cells, because there is so much wrong going on with tumor cells, including DNA damage. So this brown color reflects p53 accumulation in the nucleus of tumor cells. Normal cells here are in blue color. So that happens as long as p53 is not mutated itself, right? Because when it mutates, then it also leads to cancer. So then, when it mutates itself, then we don't see this this pattern, because there's no expression of the gene, or the expression is decreased a lot. So BRCA1 and BRCA2, those genes were discovered in early 1970s. There was there was a team in UC Berkeley that implicated a DNA repair proteins, a DNA repairing protein in breast cancer, and then, so that was 1970, and then it was a race between several groups to actually isolate, to clone the gene, and that was a consortium that succeeded, including NIH and a private company, Myriad Genetics. So they discovered, they cloned the gene that was BRCA1 as a research subject. They used an Ashkenazi family in which this mutation in this gene causing breast cancer was occurring with high frequency. So they kind of focus on a familial case there. And so the gene was found, and that led to some controversial patents, because the private company commercialized the tests. So like hospitals were not allowed to test for these mutations unless they used the assay designed by the company. So the company grew a lot. Their income was like $500 million per year. So that was really ridiculous. But these patents expired a couple years back. So now more tests are available, because this is quite simple mechanism. This is a TSG gene that may mutate in pretty much any position. So you have these various mutations showing different mechanisms involved. So it could be nonsense or frame shift, missense, or splice site mutations that inactivate the gene. So that was for BRCA1, and a year or two later, they discovered another gene, BRCA2, located on a different chromosome. BRCA1 is located on chromosome 17. BRCA2 is located on chromosome 13. And both genes have similar roles, so they function as DNA repair genes involved in genome stability. More than 500 different mutations were reported for BRCA1, and 300 different mutations were found in BRCA2. So that was a big deal, and it was one of the first, these two were first cancer-related genes identified and pretty well characterized. But it turns out that the fraction of all female breast cancer patients whose disease is caused by a germline mutation in either BRCA1 or BRCA2 genes is small and estimated between 1 and 3%. So again, mutations in these two genes, BRCA1 and BRCA2, account for only between 1 and 3% of all breast cancer cases, which is really marginal and tells us how little we still know about even such an important and common cancer as breast cancer. But when the mutation occurs, which means that the person is a carrier, the cancer risk grows a lot, and it's actually between 50 and 80% for BRCA1, and similar between 40 and 70% for BRCA2. So again, this is a breast cancer risk for female carriers. Then it also increases risk for a second primary breast cancer in females. In other words, it's the risk of recurrence. So it's 27% within 5 years for BRCA1 and 12% for BRCA2. It also increases and contributes to ovarian cancer. It also underlies male breast cancer and some other cancers as well, including prostate and pancreatic cancers. So there's a lifetime cancer risk in carriers of BRCA1 and BRCA2 mutations. So sometimes BRCA1 and BRCA2 are classified as TSGs or tumor suppressor genes, but sometimes they are classified as DNA repair genes because they are both. There are some other examples of DNA repair genes like this MSH2, which when mutated leads to colon cancer and familial sporadic types of cancer. So these genes are responsible for DNA repair, and they form this nuclear excision repair pathway, NER. There are other pathways of DNA repair, as you know, yes. Say it again. So this is loss of function, yes. So that makes them tumor suppressive genes, right? But turns out that there's a broader spectrum of phenotypic and clinical effects when DNA repair genes are mutated, and you have a short list of those. For example, xeroderma pigmentosum, it's a condition in which some of these DNA repair genes are mutated, leading to hypersensitivity to sunlight and increased skin cancer risk. Cockayne syndrome, it's also hypersensitivity to chemicals and UV light. Bloom syndrome, it's sensitivity to sunlight and associated with high incidence of leukemias, and so on. Fanconi anemia is also famous, quite famous. So there are some other, like for Fanconi's anemia, there's some symptoms that are not directly related to cancer, but at the same time, the cancer risk is increased in all these syndromes. So that's why we make this distinction between oncogenes, TSGs, and DNA repair genes, because DNA repair genes cause more problems than just cancer, okay? And like I said, BRCA1 and BRCA2 can be classified as both. Sometimes they are classified as TSG, sometimes they're classified as DNA repair genes. And we talked about epigenetics last week, and I mentioned that the role of epigenetics in cancer is of much interest, and we know that epigenetic modifications play a major role in tumor and cancer. So we know that there are two major mechanisms of or results, effects of epigenetic modifications. So one is it's like overactivation of genes and or upregulation, and the other, the opposite effect is of course downregulation, right? So these two effects have different consequences for TSGs and oncogenes. So epigenetic inactivation, when a gene is suppressed through either DNA hypermethylation or accumulation of repressive histone methylations, marks, or loss of active histone modifications, or upregulation of some microRNAs that we didn't talk about. If these mechanisms hit one of the tumor suppressor genes, that may lead to carcinogenesis. Not just ovarian is used as an example here, but it can be any cancer type. Which kind of makes sense, because here the epigenetic inactivation works kind of as a genetic mutation, right? So it's a type of inactivation. So it doesn't really matter if this is genetic through mutations or if this is epigenetic through inactivation. And the opposite effect, epigenetic derepression or upregulation, when it affects proto-oncogenes, that leads to, again, carcinogenesis. Because this is another way to produce this gain of function result, right? So this is loss of function that that affects TSGs, and this is gain of function effect through epigenetic modifications that result in oncogenesis. Does that make sense? So we talked about this effect only in the context of genetics, when there are some mutations or like gene duplications that that lead to excessive amount of the protein or some abnormally active protein produced by oncogenes. But this effect can be also produced by epigenetic changes. Yes. Imatinib is the only one that you want to remember, or this imatinib that I first introduced and then I showed you the table. So that's the only drug that you want to be aware of. But I showed you the table to illustrate the point that there is much interest in designing drugs like this, and there are more drugs already on the market, but also under development. You're welcome.